I think for the sake of time, we have to move forward to our next speaker. And I think the GENE-SWitCH project was probably the one where we had the very closest interaction within the Horizon 2020 cluster. So I'm happy that Elisabetta, representing this project, will now give a brief summary.
Okay, thank you, Krista. And well, this is GENE-SWitCH, and it started in June 2019. And as we had twin projects of this call, it elapsed for 6 months more to cope with COVID complications. And it was coordinated by myself. I am at University Paris-Saclay INRAE AgroParisTech Juin-Josas, short said INRAE, together with the deputy coordinator Hervé, and of course all the GENE-SWitCH consortium is cited here. So I started by the consortium, but of course it's thanked here. So we were 11 partners, distributed, as you see in the picture, across Europe. So it was kind of a small consortium compared to BovReg, especially, and also to Aquafang. And we had quite intense collaborations with 3 breeding companies and associations that you see listed, plus other further linked party like INSERM in France, that was a further linked party, and so on. And what was the global aim? The global aim is, of course, that we focus on 2 monogastric species that have a very high importance in animal production, bird and mammals. We wanted to deliver underpinning knowledge on their genome in terms of functional annotations, and also to enable the translations of this new knowledge for the pig and poultry sectors. And we structured the project in 3 specific but interconnected aims. The first pillar, as we called it, was made of 3 work packages, and it was devoted to carry out these functional annotations across tissues with a focus on developmental stages. We wanted to use the functional annotation for precision animal breeding. And we put quite a lot of energies also in this 3rd pillar, in aim C, for the standardization of data and processes, which was made by work package 3 and by work package 6. Aim A, it's a functional annotation with a focus on 7 tissues that are not listed here, and the 3 developmental stages, that was 30 and 70 days post-fertilization in pigs, and similar corresponding, following Carnegie tables, stages for the chicken, so 2 fetal or embryo stages, and the newborn animals. And here, what we wanted was, of course, to identify the functional elements of the genomes, characterize their tissue specificity and temporal dynamics. So from this, the name of the project, the SWitCH. We wanted to characterize by comparative genomics patterns of conservation and variation. Quite interesting to have 2 quite diverse species like birds and mammals in the same approach. And of course, we wanted to deliver open, high-quality, richly annotated genome annotations for the community. And in aim 1, just to go quite fast on some of the results we obtained in this project, I just picked some examples. About the identifications, well, we produced all these assays, but now, actually, this table that I'm showing is a bit out of date. They are all loaded on the ENA and the FANG Data Coordination Center under the Fort Lauderdale Agreement and Toronto Statement. And these were assays produced from targeted animals, plus also from animals used in the 2nd pillar from more abundant number of animals, about essentially the same assays that were tackled by the 3 projects that started together in 2019. And to analyze this data, the group, we had a kind of a single bioinformatic group from different institutions working together on all this. They made quite a lot of work on pipelines, in part in much collaboration with both REG partners, because there was a big effort on an F-core set of pipelines, with a focus, for example, in the first publication, on ISO-seq. And then there is a publication on Tagada, which is a scalable pipeline for RNA-seq analysis, and a GSM pipeline for methylation analysis. And here I pick an example of one of the analyses that actually are still ongoing, because after COVID and also for other problems, we got a big delay in the production of data. That, of course, entrained a problem of delayed analysis, where my colleagues are making now the analysis of the switches from using the different assays and then combining the outcomes of the different assays. And of course, the expectation is that this will carry out a selection, and we are just working now on trying to reduce to the most meaningful set of genes or regions of chromatin and so on. And this is just a comparison across all the tissues of the thousands of genes that actually switch between the stages, between first fetal and second fetal stage, or between second fetal stage and newborn stage. It's interesting that there are thousands, and it's much interesting, I think, is that most of these genes are tissue-specific. So there is quite a lot of information there. And the same pattern, essentially, is seen for chromatin accessibility. And here you see, for example, there are tissues like liver, where on average you have less genes that are moving. You see that both in the chicken and both in the pigs, for example. And then some tissues, like, for example, skin, where you have really thousands, quite a number of genes that are moving. As I said, it's just an example of the many that are going on to analyze this set of data. The other thing we produced in this aim was a first ensemble regulatory build for different pig breeds, not just salmon, and both the broiler and white leghorn plus the jungle fowl reference chicken. We are quite proud of that because we contributed to ensemble, as also Aquafang. In this picture, you see also the contribution of Aquafang. The picture is a bit outdated now because the last ensemble release is from this January, and some annotated regions were relabeled for enhancers and also based on the overlap with the Eastern Chipsic peaks that we obtained very late in the project. The aim B is about using functional annotation for precision animal breeding. And we took two very different approaches. The first one, the so-called genomic selection one, it was based on the main question, can functional annotations enhance the prediction accuracy of breeding value in commercial population? And the second one was a kind of exploratory approach. At least at the time was very exploratory because, we talk of 2018 minimum when we had really to finalize the project. So it was to provide the basis for future studies focused on better farm management. So something complementary to genomic selection and aimed to pave the way to use a lower quality and more sustainable feed, rich in fibers in this case. So going to the first approach, this was work package 4, which so far has been the biggest of GENE-SWitCH work packages. And the aim was to extend genomic prediction models to exploit the new annotation maps in both species. And they did this by a number of approaches, especially in particular by developing new genomic prediction models, in generating new data sets, to find map QTLs and in chicken especially, and by a EQTL study made in pig. And then the last and big part as well, that took most of the last period of the project, was validating these outcomes of these models in large scale commercial data for both species. The main outputs, I said here some of the papers that are already out. There have been really important focus on the model development, both on Bayes-Co RCO software that is now available, but in general using Bayes models and machine learning models, with some interesting insights, like that the machine learning models seems to better explain phenotypes for epistatic traits compared to other models. Interesting that the gene expression explains considerable variance depending on the moment of sampling, which is, of course, also relevant also for potential use in real breeding programs, given also the cost of this kind of approach. For QTL mapping, we published EQTLs in pigs and fine-mapped QTLs in chickens. That is under publication as well. And concerning the validation of genomic prediction in commercial populations, there was a very recent and interesting results, in which FAPE scores, which is functional and evolutionary trait heritability scores, which were actually first discovered in bovine, I think, in 2018 by Xiang et al., have been applied. And they found that this combination of different layers of annotations improves accuracy in pigs. 50 minutes, right? I will say some words on these epigenetic effects of maternal high-fiber diets in utero and the offspring. This was the other side of this approach. We wanted to see to assess the epigenetic effects of maternal diets that varies in fiber contents on pig fetuses and weaned piglets. We achieved this by dedicated in vivo experiment, where we produced two progenies in pigs, by the analysis of differential transcribed genes and chromatin accessibility regions, and by integrative analysis that included microbiota composition, short-chain fatty acids measures, and weight phenotypes. I'm not giving any background of this, but the idea is that you have a microbiota, and you have metabolites produced by a microbiota that vary in function of the diet that you take, and especially in the fiber that you take. And many, actually not most, of these metabolites go into circulation and could affect the epigenetics of peripheral tissues, as demonstrated in mice. As main outputs, we found that actually there was a pervasive alteration of the functional genome of liver and muscle tissues. And this effect was better detectable in piglets than in fetus. I was surprised, but they said they should not. Anyway, because piglets were all on the same diet after weaning. Other things are not really not significant, but what we found of interest was an evidence of anti-inflammatory phenotypes in both liver and muscle triggered by high fibers. This is something which is now being published. I will say also one word about AMC, because we had a lot of activities here for the coordination, standardization, validation, curation, and archiving of all the data. The clustering activity, as Krista mentioned also, we interacted quite a lot with the H2020 project, and we made the start of Eurofund. And we made also a number of activities for implementing training, capacity building in both academia and the industry. Up. To finish, did the GENE-SWitCH match expected impacts? I think overall, yes. We could always do better, huh? But anyway, we'll see. We deliver a high-quality reference annotation map that impact on several areas, essentially on any area of fundamental science, and in applied science as well, because we are also important for the animal industry specialists as a reference. New genomic prediction models have been delivered and tested, and this has gone with publications and papers in preparations and in press, but also with a lot of activities to disseminate this to industry. We paved the way for follow-up investigations. There is GENomE, a project that is now at the second year, which is also focused on chicken and pigs, and also for nutritional studies. And we have co-funded Eurofund that provides great opportunity for large-scope G2P research in Europe and in the next decade. All about us is here, because the website has been updated, so you find everything, also from our final conference that was in last November, and was as well this one a 10 years event. And the final thanks to all participants, Eurofund collaborators, Bovary Consortium, and thank you for your attention.